Good morning, students. This is your teacher Neelam Helawat, and today I have come up with the last topic, and that too very important topic. That is your DNA fingerprinting. I hope you are familiar with this word. You've heard this name every time, isn't it? It's a very interesting topic, right? And even a common man has heard this name, DNA fingerprinting, right? And this is the last chapter of this topic. So with this topic, we'll be finishing this chapter, right? And I hope so far you have understood all the concepts well, right? The transcription, translation, right? Genetic code and the human genome project. I hope you're well versed with the concepts of these topics. Even then, if there is doubt, students, you can talk to me. You can send your queries to me. I can discuss those queries with you, right? So let's start with the today's topic, that is DNA fingerprinting, and it's a very important topic, everyone. Are you ready? Okay, let's start then. So DNA fingerprinting is basically a forensic technique, right? This is basically a forensic technique which is used to identify the individuals by the characteristics, that is of their DNA, right? This technique is extensively used in the forensic science, where there is a debate regarding the criminal cases, or there is a parental dispute regarding the child. So this technique is extensively used. Now this DNA fingerprinting was invented by a scientist named Alec Jeffrey. The name of the scientist is Alec Jeffrey, and he belonged to the University of Leicester in 1985. So he discovered this technique. He is the inventor of this technique, and this technique is also called as DNA profiling or molecular fingerprinting. See, DNA fingerprinting is a very common name. As I told you, even a common person understand what is DNA fingerprinting. What do you mean by this DNA fingerprinting? But it is also known as DNA profiling or molecular fingerprinting. Right, everyone? Now, basically, we'll talk about what this is, what are the principles, on which principle this technique works, how it is done, what are the applications, everyone. So, where this technique is particularly utilized. So, this particular, you can say, topic is very important from the exam point of view. So, we'll go through each, you can say, each paragraph or each topic separately, since this technique is very important. So first of all, what is a principle? How this technique works, right? The DNA, the small parts of the DNA that vary from individual to individual, right? As we know that approximately some percent of DNA, that is, I think 99% of the DNA is similar, isn't it? As we've discussed yesterday, 99% of the DNA is similar. But there are certain parts of the DNA that are different from one individual to different individual, right? And these are referred to as variable number of tandem repeats. What do we call them as? Variable number of tandem repeats. What are variable number of tandem repeats? Hanji students, tell me, what is the variable number of tandem repeats? What are they? They are the small part of the DNA. They are small part of the DNA that vary from individual to individual, right? Because we know 99.9% of the base sequences in humans is same, right? And what is the genome size? Yes, everyone, can you tell me what are the genome size in human beings? How many base pairs we have? We have 3 into 10 to the power 9 base pairs. We exactly have 3 into 10 to the power 9 base pairs. And out of this, as I told you, 99.9% of the base pairs, they are similar in each individual. So all human beings, they have this much percentage similar. So there is only a small fraction of DNA that will be different from one individual, one human being to another human being, right? And that is called, that particular portion of DNA is called variable number of tandem repeats. Okay, that is called variable number of tandem repeats, right? Suppose you have been seen here this chromosome, it's a paternal one. So you can see these are the tandem repeats. These portions, they indicate the VNTR. Okay, so we can compare these VNTRs, right? We can compare these VNTRs, that is the portions of DNA that are different from each other, right? Now, how do we go about this technique? What is the methodology of this technique? What are the various steps that we follow during this technique? So, first of all, the cell's sample is taken. So, first of all, we will take a sample. Now, this sample could be taken as a blood sample. We can have blood from the particular person. We can take a hair follicle rather. We can take a small portion of the skin. We can take a small portion of the bone also, saliva also, semen also, right? We can take semen, small portion of the semen, a sample of the portion. So, we can collect any of these samples. So, first of all, sample collection. We will first collect sample from the individual. Now, once we have collected the sample, the cells are broken to release the DNA. Now, suppose this is your sample of DNA. So, we have taken the DNA sample, or you can say blood sample we have taken. Blood sample is the easiest sample that is generally taken in case of the human beings, right? So, the cells will be broken to release the DNA. We know, dear students, that cell has nucleus, and inside the nucleus your DNA is present. So, first of all, these cells, they will be broken to release the DNA. This is your first step. And this step is called extraction of DNA, right? So, we will break the cells and will obtain the DNA. The second step is digestion of DNA by restriction endonuclease. So, the DNA that we have obtained, suppose this is your DNA, right? The double helical structure, we have obtained DNA. Now, this DNA will be cut. This DNA will be cut into small fragments by an enzyme which is called restriction enzyme, right? This enzyme will cut the DNA and it will form small segments, right? Like this, you can see we have shown a sequence in the, just look at the picture, I have shown a sequence, like this is your double helical structure. So, this green portion is being cut by the restriction enzyme. So, DNA is a very, very big molecule. So, first DNA will be converted into small fragments, and this breakdown of, or you can say segments will be small segments of DNA will be made by an enzyme which is called restriction enzyme. Okay. The third will be these fragments, they will be separated by length. Now, restriction enzyme has made small fragments of the DNA, right? These small fragments, they will be separated by their different length. Okay. We know the DNA is negatively charged, isn't it? So, it will move towards a positive terminal. So, we will connect it with this device. So, this is a positive part of the battery and this is a negative part. Since we know DNA is negative, so it will move towards the positive end. Okay. And the shorter fragments, they will move faster. So, what will we do? We will pour the DNA fragments on a gel bed. And this gel bed is made up of agarose gel. So, this is your sheet. From here, we will be putting the samples and the DNA will start moving, isn't it? The small fragments, they will start moving and the shorter fragments will move faster. So, on the lower side, you will find very, very short fragments, and then the upper side, you will find the bigger fragments, and on the top, you will find the longer fragments. Okay. So, third step is separation of DNA fragments by electrophoresis. So, this technique that we will use to separate the smaller fragments, or we'd say we will arrange the smaller fragments serial wise, smaller to the larger fragments on agarose gel bed. So, this technique is called electrophoresis, right? Now then, what will we do? The DNA fragments, they will be transferred from the gel to the filter paper or a nylon sheath. Okay, they will be transferred to the nylon sheath, and DNA is split into single strand using an alkaline solution. Right, everyone? Now, suppose you have a gel. So, from the gel, right, we will transfer it on the tissue paper or we will transfer it on a nylon sheath. We can use any one of them, right? Now we will transfer, and then what will we do? We will split the DNA. We know DNA is double-stranded, right? DNA is double-stranded, so we will separate the two strands of the DNA by using an alkaline solution, right? See, we have separated. We can easily look at this diagram. This is your double helical structure DNA. Now this green portion is your alkaline solution. This alkaline solution, this will separate the two segments of the DNA. Okay, so two segments of the DNA, they have been separated. Now we got two portions of DNA, right? Now we will use radioactive probes. Okay, we will use radioactive probes that will bind to this DNA. Okay, so first is हमने जो DNA fragments obtain किए हैं उन्हें हम नायलॉन शीट पर या फिल्टर पेपर पे ट्रांसफ़र करेंगे. उसके बाद हम एक अल्कलाइन सोल्यूशन यूज़ करेंगे जो DNA को सेपरेट कर देगा, अलग अलग फ्रेगमेंट्स में सेपरेट कर देगा. उसके बाद हम एक रेडियो एक्टिव प्रॉब यूज़ करेंगे जो DNA से बाइंड कर जाएगा. ओके, दैट विल बाइंड विद द DNA, राइट? Now once this has occurred, now we'll place an X-ray film on over the filter paper. अब हम एक X-ray film रखेंगे filter paper पे, and these radioactive probes will be marked on the screen, right? So we'll keep this X-ray on the filter paper. So once we put it on the filter paper, we will obtain certain marks, like this kind of marks will be obtained. These are nothing but they will be called as DNA fingerprinting patterns. They are referred to as DNA fingerprinting patterns. Okay, everyone, they are referred to as DNA fingerprinting patterns. Okay, so in this manner, we will develop the DNA fingerprints of the individuals. If there are two individuals, then we will have two X-ray films, right? We will compare these two X-ray films with each other. If their repeats match, then obviously you can easily, if it matches, then obviously there is a solution to your question, right? So two persons are there, we will prepare two X-ray films and we will have two DNA fingerprinting patterns, and then we will compare to these two DNA fingerprinting patterns. Okay, everyone? Now this technique is extensively used in the murder cases, right? So from the crime scene, the samples will be collected and then they are matched with the criminal record, and then it is easily to detect who has done this crime. Then the forensic team, basically, you know that in the police department there is also a forensic team. This forensic team will trace the blood sample from the crime scene, and then they will prepare the DNA profiles, and then these DNA profiles, they are matched with the victim and the suspect, and if they matches, then obviously your suspect is the criminal. Apart from this, it is also this technique is also used to establish the individuality of a person, right? This is also used to solve the problems of paternity and maternity disputes, like the child is whether biological child of the two parents or not. So such disputes, they can also be solved. ठीक है, ये बच्चा किन पेरेंट्स का है, ये ऐसी जो प्रॉब्लम्स हैं, they are also solved with this method. Then it can also use to detect the heredity problems, right? And it is also used in the forensic, right? Then sociology also, some of these techniques are also used in sociology for the social cause. So this technique is extensively used in the medical science, and we refer to as a forensic science rather, and it helps to solve a lot of problems, right? It helps to solve a lot of problems, but this DNA fingerprinting is a legalized technique. It can only be conducted when under the permission of a Supreme Court. So no hospital can perform it as its own. So it is under the legal control. This technique is under the legal control. Okay, everyone? Otherwise, it could be used, you can say, it will become a pen and penny for everyone, right? So everybody will start using it. So this technique is not used very commonly, but yes, it is used in case of debatable issues. Okay? So coming to homework, so you will make notes of this topic and you will read the page numbers 121 and 123. Okay, and students, you will also complete your NCERT questions of this chapter. And I suppose with this topic, we have completed the chapter 6 also. And from tomorrow onwards, since you've got your date sheet, we will start revising our lessons. So if there is any definite, you can say definite portion of the syllabus or definite chapter that you want me to revise, so you can send those topics to me. I'll definitely try to resolve your problems by making such videos. And please do like the videos, do share them, because that really motivates me if I get an appreciation of my work. Right, everyone? So that's all. Bye, everyone.